In chapter 11, we switch gears a little bit and we're going to talk about bacteria and viruses. Bacteria do not reproduce sexually, so we've been talking about how genes are passed down among organisms using sexual reproduction, but bacteria don't do this. So we're going to talk about how bacteria actually share genes and pass variation among their populations. We'll also talk a little bit about viruses and their role in this transfer of genetic information. So the advantage of sexual reproduction is that it produces an exchange of genetic material. And when it does this, it produces a huge amount of genetic variation. We saw crossing over or recombination where chromosomes physically exchange information. We saw shuffling of chromosomes in random assortment where you get new combinations in the offspring. And then we saw random fertilization where you get an unrelated egg and an unrelated sperm coming together to produce even more genetic variation. But the vast majority of organisms on Earth actually produce via asexual reproduction. So we've been focusing on our diploid organisms, basically our plants and animals. They're diploid or polyploid. A lot of plants are polyploid. And this means that they are sharing genetic information with one another. They have multiple copies of chromosomes that come from each parent. But most of the organisms on Earth are actually bacteria and archaea, which reproduce asexually. Asexual reproduction produces clones, and when you produce a clone, you're essentially making a copy of yourself. This could be likened to mitosis, where one cell divides into two genetically identical cells. The same thing's happening in bacteria. When you have asexual reproduction, you have no shuffling of genetic information. You have no variation. But genetic exchange actually does still occur in bacteria and is really important in bacterial evolution and in bacterial response to their environment. So there are three ways in which bacteria take up or acquire new genetic material. One is transformation, where they take up DNA from the surrounding environment. One is conjugation, where there's actually a physical connection between two bacteria and one transfers DNA to the other. And the other is transduction, where viruses actually infect a cell with another cell's DNA. When we're talking about producing offspring, we are doing something called vertical gene transfer. This is where you have a parent, and that parent passes its genetic material to an offspring. So essentially, DNA is transmitted from one generation to the other. And when you're doing vertical gene transfer, you're going to produce clones. These clones are going to be basically identical to their parents, if not identical, very, very similar. You might have some mutations that produce new alleles or polymorphisms in the population, but any variation is going to be very slight. So this is represented here by slightly different color blues. You might have offspring from the same parent that are just a little bit different, but very, very similar. And so if you look at the population, you have a lot of identical individuals, and even if they're not identical, there's still very slight variation. We can also have horizontal gene transfer. So horizontal gene transfer is basically transfer of genes from one population to another, not from generation to generation. So in this diagram here, we have the orange population transferring a small piece of DNA to the blue population. And when this happens, it creates genetic variation within that blue population. And some of these blue individuals now have new information. They've acquired new information that allows them to be different than their surrounding cells. So let's pause real quick and review bacteria just very quickly. So bacteria have a plasma membrane. They have a larger circular piece of genomic DNA. They do not have organelles, but they do have ribosomes. So these ribosomes are the site of protein production. So they use the genomic DNA to produce proteins. Now this is different from, this situation is different from eukaryotic cells where we have multiple linear chromosomes. Bacteria also have what we call plasmids. So plasmids are small circular pieces of DNA. There might be a couple or there might be thousands in a particular bacterium. And plasmids are what we call transposable. That means that they can be transferred from one organism to another. And very often within a population of bacteria, it is those plasmids that are transferred. And these plasmids often produce some sort or code for something that is important in the environment. So this might be antibiotic resistance, which is how resistance can spread through a medical environment. It might be the ability to utilize a particular nutrient in the environment or to deal with some sort of stress within the environment. Now, if the organism is not using these, it's not beneficial for the cell to maintain them, to replicate them, and so they can be lost as well. But they can also be transferred horizontally. Now, horizontal gene transfer can happen using happen to plasmids, which is what's being depicted here. But you can also transfer genomic DNA. So some of this genomic DNA can actually be transferred horizontally as well. So in horizontal gene transfer, individuals from different populations within the same generation level can exchange DNA. Now, this horizontal gene transfer is going to increase the variation within a population when that information is exchanged between different lineages. This is going to produce new combos of alleles. It might even introduce new alleles or new polymorphisms into the population, or it might produce entirely new genes that have been acquired from the environment or from another type of bacteria. And this is really important in increasing variation within a bacterial population. So this increase in variation is really important because it allows that bacterial population to respond to their environment. So let's say that this orange population encodes some sort of antibiotic resistance. And this one over here, the blue, doesn't. They have no resistance. So in the presence of an antibiotic, these blue organisms are going to be wiped out. They're going to be killed by the antibiotic. But let's say that this orange population happens to come in contact with the blue population and the gene is transferred horizontally from one to the other. Now, these organisms that have this new information are now going to encode that antibiotic resistance as well. And so while this is not beneficial to the human population, it's beneficial to that bacterial population because they have acquired this new variation that has allowed them to compete within their environment. So in eukaryotic reproduction, we get gene transfer that basically happens between two cells. Basically, those cells are coming together. They are both donating their DNA. But in horizontal gene transfer, DNA is transferred unidirectionally. That means that there is a recipient cell and there's a donor cell. Doesn't go both ways. So if that donor cell donates a piece of DNA to a recipient, that new DNA is likely to be expressed in the recipient cell. Now, one of two things can happen. That free DNA that has come in can be integrated into the genome, which is pictured here. And when it is integrated into the genome, it's going to be expressed just like the other genes within that cell. that DNA may also be maintained as a plasmid. So if this piece of DNA that's being transferred is actually a plasmid, let's say that it's a plasmid, it can be maintained as a double-stranded circular piece of DNA within the cell as well. If it is integrated into the genome, if the piece of DNA is integrated into the genome, then it is going to then be transferred vertically. That means it's going to be transferred to all the offspring. The plasmid, if it is maintained in the cell, may be transferred vertically as well, but not necessarily, because that plasmid can be lost. But once it is integrated into the genome, whether that's plasmid information that enters the genome or genomic DNA from the donor that's integrated into the genome, once it is integrated, it's going to continue to be passed on. It's going to be part of that coding DNA of that bacterium. Now, this imported DNA can be detrimental. It can be detrimental to the organism. And so the uptake and integration of foreign DNA is actually tightly regulated by the cell. They aren't always maintained. Remember, we have talked about restriction enzymes that actually break down infecting DNA. And we'll talk about CRISPR in just a few minutes, or in another video, which is another mechanism by which bacteria actually essentially make an immune response to invading DNA. So we've talked a little bit about plasmids. We've actually seen a plasmid in our lab so far. We saw the P-glow plasmid. Whether your class did the P-glow transformation or just the P-glow plasmid restriction, we actually saw that plasmid. This is another plasmid. This example plasmid is called PSB 102. This is a fairly large plasmid, 55,000 base pairs. And you'll see that it encodes a variety of genes. This particular plasmid encodes a set of genes that give it an environmental advantage. It allows it to live in a special environment. These genes actually allow it to be resistant to mercury in its environment. So mercury in an environment is normally detrimental to living cells, but this particular plasmid encodes genes that allow it to cope and be resistant to that mercury. So here's our example of one of those beneficial survival genes or beneficial environmental genes. So plasmids vary widely in size. The P-glow plasmid that we saw in lab is only about 5,000 base pairs, so it's one of the smaller ones. The gene composition can vary as well. Plasmids can be maintained, I believe I said earlier, in small numbers or high numbers, depending on what they encode and how beneficial they are to the organism. Just before we go from here, these green genes are involved in transmissibility. So this means that the plasmid actually encodes genes for transfer. So it actually encodes genes that direct the transfer of this plasmid from one organism to another, and we'll talk about that in just a minute. So this example gives an environmental advantage. We talked about antibiotic resistance. Sometimes these plasmids can encode a ability to use a particular nutrient. But as I said, they're going to be maintained as long as they are beneficial. These plasmids are known or are called replicons because they actually replicate separately from chromosomal DNA. So they perform their own replication. So basically, they maintain themselves. They direct their own replication separately from the chromosomal or genomic DNA. So you might notice these arrows that are on our DNA molecule here. Those indicate the direction of the gene. you see that genes can run in both directions. They are encoded on either strand. You've got some different directions going on here, and we'll get to this a little bit later as well, but genes can also be encoded on opposite strands running opposite directions. So there's a lot of room to encode many, many genes within even just a small piece of DNA like a plasmid. All right, so let's talk about these different types or different mechanisms of genetic exchange. So we'll talk about transformation in this video and the other two in the next video. So the process of transformation occurs through the uptake of DNA from the environment. So any cell can actually take up DNA from the environment. Some eukaryotic cells do. Some cells need to be forced to take it up, and you do this by shocking the cell in some way, providing a particular chemical or heat or something that makes it competent to take up DNA. Oops, I spelled that wrong. So a cell that has the ability to take up DNA is going to take it up and pull it into its cell. Once in there, that piece of DNA can be converted and inserted into the genome. So one single strand is going to be taken up. And as the piece of DNA is taken up, the single strand is actually degraded and not taken into the cell. So only one strand is actually taken up into the cell. Now, cells do not very readily take up DNA. And in fact, very few cells can actually be made to be competent. So even among billions of bacteria, very few will actually be transformed. Once that linear single-stranded piece of DNA is taken up, that DNA is replicated by machinery within the cell and made double-stranded. Once it is made double-stranded, it can be integrated into the genomic DNA. Once it is double-stranded, it's more stable and can be incorporated into that double-stranded piece of genome. Very frequently, transformation is done in the lab. And when you do this, you are able to make cells that contain recombinant DNA. So recombinant DNA is DNA that contains information from two sources. So we might want to, in a laboratory, actually get our E. coli or other bacteria to take up and express a piece of DNA from another organism. Now, there's a variety of reasons we might do this. We might do this to study the DNA, to study the gene, to see what protein it makes, or to see how it functions. We might use this for pharmaceutical purposes. For example, insulin is inserted into E. coli cells and the E. coli are used to actually produce insulin for medicinal purposes for treating diabetes. And there are a variety of other biotechnological techniques that we could use transformation for. Depending on which semester you are in, you may do the pGlow transformation lab, which is where we transform E. coli with a firefly gene or a jellyfish gene that allows the bacteria to grow. So it actually glow under a UV light. It actually shines that fluorescent green color that you're familiar with from fireflies. If you are in a semester that does not do pGlow, you will probably be doing the LAC-Z transformation. This allows a cell to express a gene that turns the colony of bacteria either blue or white, depending on the presence of the gene. So these are both used as indicators to make sure that the bacteria has taken up the gene of interest. You know that if it is producing this glow or producing the right color, that it has actually taken up the plasmid. So in a lab, you might have a plasmid that has your gene of interest and an indicator gene, so that you know that your gene of interest has actually been taken up. But in a natural situation, bacteria can take these pieces of DNA up as well. And this is how we get transfer of genetic material, such as antibiotic resistance in those plasmids. So if we're going to use plasmid for laboratory cloning, essentially laboratory cloning is using a bacterial gene to make copies of the gene of interest. So I mentioned LAC-Z gene. There's actually a box in your textbook, box 11.1, that talks about using this LAC-Z gene for blue-white screening. So basically, in this image here, we are introducing a particular gene over here at what we call the multiple cloning site. This is just a site that can be cut with any one of many restriction enzymes. So we can cut our gene of interest with a particular restriction enzyme. So we've got restriction enzyme site on both sides. We can cut our plasmid with that same restriction enzyme and allow them to anneal or come together. Now, when we do that, we actually interrupt this LAC-Z gene. Another important gene that we have in this plasmid is ampicillin. This gene provides resistance to ampicillin, which is an antibiotic. And this allows us to select for our bacteria that have taken up our plasmid. So when transformation has successfully happened, we have cells that can survive on plates with the antibiotic ampicillin. By inserting our gene into this LAC-Z gene, we can actually use that blue-white screening. So any cells that have taken up the plasmid are going to be resistant to ampicillin, and any cells that have the plasmid with our gene inserted are going to interrupt this LAC-Z gene. So the LAC-Z gene is able to cleave a chemical called X-gal. It's a sugar. And when this LAC-Z gene produces the protein that breaks down that X-gal, the bacteria are going to appear blue. When that LAC-Z gene is interrupted by our gene of interest, that plasmid does not encode a functional LAC-Z anymore, so it doesn't produce the blue color, and instead our colonies are white. So there are two mechanisms that we can use to determine that we have inserted our plasmid and then also inserted the plasmid with the gene that we wanted. Because remember, not all cells are going to take up DNA, and not all of our plasmids are actually going to get the gene. So this is sort of a two-step process. We're going to insert the gene, and then we're going to insert the plasmid into bacteria. So if we have a bacterial colony that can, one, grow on ampicillin plates because it has ampicillin resistance, and two, shows white colonies, then we know that our cell has our plasmid that we want with our gene of interest. And then we can go on with our recombinant analysis. We can do whatever it is that we're going to do with this plasmid. So the rest of this slide just shows an expanded view of that multiple cloning site where we have our restriction enzymes. So if let's say you were to cut this PUC18 plasmid with PST1, this is one that we saw in one of our labs, you would make your cut here. I don't know exactly how it PST1 cuts, but you would cut your PUC18 plasmid with PST1. You would cut your gene with PST1, stick it in, allow them to come together, and now you have your PUC18 plasmid with your gene of interest. So I've added a couple of slides. These slides are not going to be on your probably not going to be on your study guide because I added them in fall 2020. If they're not, please pay attention, make a little note on your study guide to go back and review these. So let's say that we want to use one of these recombinant plasmids for some biotech purpose. If, let's say, we want to introduce a gene into a eukaryotic organism because we want to make a transgenic or genetically modified organism, we'll give an example in just a minute. Let's say that we want to make a genetically modified organism. How are we actually going to get that gene of interest that we put into our bacteria into the genome of our cell? Well, once that foreign DNA is inside of a host cell, there are mechanisms already present in that host cell that are actually going to, quote unquote, repair the problem. And they can mediate the recombination, just like we saw in chapter 6 with mitosis meiosis with crossing over, and in chapter 9 with our mapping. These proteins can actually mediate a recombination that allows our DNA to join the foreign DNA to the host cell's chromosomes. So let's say we have a eukaryotic cell. I'm just going to draw a nucleus here. And we have our little E. coli cell with our plasmid of interest. And then we've also got our genomic DNA. I'm actually going to draw our plasmid in a different color. Here's our plasmid. We can actually, if this is a eukaryotic cell, use the bacterial cell to infect our eukaryotic cell, essentially allow that plasmid to enter the eukaryotic cell, and then these repair and recombination enzymes can actually use a sequence within our plasmid to recombine with the eukaryotic genome to actually express the gene where we want it to. Now, this will happen at a place that has a similar sequence between our plasmid and our eukaryotic genome. So let's say that we want to express a particular gene in a eukaryotic cell or even in another bacterial cell. It doesn't have to be a eukaryote. Or let's say that this is occurring in a natural population and a plasmid has entered a host cell and this gene is going to be incorporated into the genome. This transforming DNA, this DNA that's coming in, has to have a sequence that is similar to the host genome. When we talked about recombination in eukaryotic cells, homologous chromosomes line up because their sequences are similar, because their sequences are the same. So this transforming DNA works on the same principle. It has to have a sequence that is similar to the host DNA. So this brings up something that's important for bacteria, in that transformation generally occurs between similar or related species that are already similar, that have some similar DNA. Because when they have this similar DNA can line up and cross over. And then crossing over happens just like we saw in our other chapters. So our homologous pieces of DNA are going to line up. They're going to perform crossing over. You require a double crossover event in order to actually incorporate that transforming DNA. And so when this happens, you get your new DNA exchanging information with your old. Your host DNA exchanging information with your new. And you get this new piece of transformed DNA incorporated into the host genome. And now that new piece of DNA can be expressed just like the DNA that is already being expressed in your organism. So if we're doing this for some sort of laboratory purpose, we now have our host DNA expressing this DNA that we want to do something with. Maybe this is an E. coli that has taken up a gene that we want to work with. We want to have the E. coli maintain this gene so that we can study it. We can do the sequencing. We can analyze expression, things like that. Or maybe this is a gene that we are performing this biotechnology, this transgenic organism production. I'll give you an example of that in just a minute. Or maybe this is bacteria who have taken it up from the environment and now they have new genetic information. We now have new variation in that host bacterium's genome. So I wanted to do a quick example of how we might use this to make a genetically engineered organism or a transgenic organism. So a genetically engineered organism is any organism whose genome has been modified, and this is going to be modified for a special purpose. Generally, this is going to be modification sort of in an unnatural way to be able to engineer this genome to do something different than it has previously done. We can also make a transgenic organism. So this is an organism that has genetic material from a foreign source. Okay, so they're slightly different. Transgenic organisms are genetically engineered or genetically modified. GMO, that's kind of a buzzword, genetically modified organism. But not all genetically engineered organisms are transgenic. So just a quick review from a couple slides ago. Our gene of interest is going to be removed using restriction enzymes from the genome of the organism we want to take it from. It's going to be inserted into a plasmid using the same restriction enzyme. That plasmid is going to be transformed into host bacteria. And then let's say that we want to make something called Bt corn. Bt corn. So Bt corn is a type of corn that has been engineered in order to produce these little crystals within the kernels that actually deter pests. So basically they are this is pest resistant corn. So let's say we want to make this pest resistant corn. We're going to allow that bacteria that we have transformed to actually infect the host cell, to infect the embryos of this corn, the baby corn, so that all of the cells in this corn produce this pesticide resistant crystal. Once that bacteria has infected the host cells, recombination is going to allow that gene to enter the host cell's chromosomes. And then that host cell is going to express our gene for pesticide resistance. I'm sorry, pest resistance, not pesticide resistance, pest resistance. So here's an example. This actually comes from my Bio 100 textbook that we use in that class. But I think it's really interesting. Our book doesn't give an actual example of an application of this plasmid use. They tell us how we can use them, but they don't tell us how they are specifically used. So this corn is engineered to contain a gene from a bacterium called Bacillus thuringiensis, and that bacterium naturally produces a little crystal that actually kills insects when they eat it, but doesn't harm humans. So this Bt crystal gene is actually expressed in the corn cell so that when insects like caterpillars eat their leaf, they're killed. So this helps to prevent insect predation. The advantage of this is that pesticides are no longer needed, so we don't have to spray pesticides on the crop. Now we can use this Bt crystal gene because it doesn't harm humans. It doesn't have any effect on us, just on the pests that are infecting this plant. So just sort of a quick recap, because I went a slightly different direction than our book. In the process of transformation, bacteria take up DNA from their environment. Now, this is a natural process that keeps genetic variation within a bacterial population because it can acquire these new alleles, new genes, new genetic information from the environment. But we can also use this in a lab, use this same idea in order to introduce genes from other organisms into a bacterial cell for a variety of purposes. Now, this works because this process of gene expression is the same or very, very similar in all organisms. And all organisms on Earth use the same process of gene expression. So this gene from this bacillus bacteria is going to be expressed in the corn cell just like it would in the bacterium. And so this whole process, gene expression, which if you remember is called the central dogma, the process of transcription and translation, is the same or really, really similar no matter what organism you use. And so we can successfully transfer genes between living organisms.